genome editing therapies for treating rare diseases one time per lifetime. Okay, so we address a big group of rare genetic disorders that are called haploinsufficiencies. If you're not familiar with it, just know that each gene has two copies. One comes from the father and the other comes from the mother. If one copy is broken and the other one cannot produce enough protein to sustain the normal function of the gene, then an haploinsufficiency will occur. Just let me give you some example. If the copy of the OPA1 gene is broken, then you will develop the autosomal dominant optic atrophy. And keep in mind this because I will need it later. If the copy of NCF1 gene is broken, then you will get an immune system disease, and so on and so forth. There are 300 of these diseases, and 99% cannot be cured, and they are manageable only with palliative and symptomatic care. So there is very little to offer to the 50 million patients that need a therapy that cure haploinsufficiency in a single dose treatment. With our technology, we are doing genome editing, and thus we are installing precise and permanent nucleotide modification in the genome of the patient, and those modifications are able to boost the translation of the haploinsufficient gene, and thus boosting the amount of protein that is produced in the cells that we edit. With our technology, we are able to precisely increase the protein expression of the haploinsufficient gene in order to reach a physiological protein level that will cure the disease. Now, I'm showing some data in which we edit cells and disease-relevant cells, and we show that the mRNA transcription level stays the same, but we were able to boost the protein level of the NCF1 gene that causes an haploinsufficient disease. And this increase varies depending on the type of modification that we install. And thus, our therapy can be personalized for the patient needs in terms of gene expression. So how it works, our therapy? Well, we deliver the genome editors inside the tissue that is affected by the disease, and just that one. Then we install precise nucleotide changes in the haploinsufficient gene, and those changes are able to increase the expression of the target gene. We edit all the possible changes that can boost the translation and rescue haploinsufficiencies. But now we want to validate our platform with the target disease. And the target disease is ADOA. As I mentioned, it's a disease that affects the eye, and the eye is an organ in which we can easily target with the delivery system, and thus delivering our genome editors. And also ADOA has very high prevalence in Europe, as in 1 in 30,000 people has carried the disease nowadays. The market for haploinsufficiency is very wide, and as there are 50 million patients that carry these diseases. But first, we want to start addressing the European and the US market with our EditUP platform, and starting specifically from the 25,000 patients that carry the ADOA disease. So we have two groups of competitors: those who are using the strategy of boosting the transcription with mRNA or other approaches, and they are boosting the genes, the haploinsufficient genes, such as OPA1 and others, and thus they have a platform technology like ours. But we have a really personalized therapy for the patient such as our other competitors that use another strategy that is called gene replacement. Very simple, they take the cell from the patient, they deliver the corrected gene, and then reimplant the cell into the patient. This strategy is for sure personalized, but it's not precise. In fact, they do not know where the actual functional copy is integrated into the genome. And finally, our first characteristic, we aim to provide a cure in a single treatment, unlike our other competitors, first competitors that will need recurrent treatment of the patient. So we filed the patent, and in the next couple of years, we want to validate our technology for the EDOA disease with in vivo and in vitro assays. Next, we want to get the license for a genome editor and the delivery system and run the preclinical phase thanks to a partnership with the CRO. Next, we will do the clinical trials in which we will have multiple licensing opportunities, both for EDOA, but also for other haploinsufficient diseases. We are planning to reach the market in 2031, and after carefully reviewing the markets based on the cost of the therapy, gene therapy, and the number of patients, we are planning to get millions in revenues coming from royalty payouts. So to wrap up, we are EditUP genome editing startup. We are aiming to provide a solution, lifeline solution that is safe, personalized for the patient that carry haploinsufficiency, first validating the autosomal dominant optic atrophy. Okay. So I'm Alberto Rao. I'm doing the research part, and I'm supported by the University of Trento, and Davide Ederle is helping me develop this startup. Thank you. Thank you.